Hello students, in this video we will discuss 30 important questions from chapter number 10, cell cycle and cell division. Question number 1, minimum how long does the cell division proper last in a human cell and what percent of duration of cell cycle does interphase last? In human cell division lasts for about 1 hour out of total 24 hours of cell cycle, and interphase lasts for more than 95% of duration of cell cycle. Question 2. Name the last stage of prophase 1. What marks this stage? The last stage of prophase 1 is diakinesis, and it marks the terminalization of chiasmata. Chiasmata is a point where exchange of genetic material takes place between homologous chromosomes. You can see in the diagram the displacement of this chiasmata is taking place, and it is called as terminalization of chiasmata, which occurs during diakinesis of prophase 1 meiosis 1. Question 3. What is interkinesis? It is a very short stage between meiosis 1 and meiosis 2. Question 4. Name the stage of cell division in which chromosomes cluster towards poles. In telophase stage of cell division, the cluster of chromosomes are present towards the poles. Check these cluster of chromosomes at poles in diagram also. Question number 5. The phase of nuclear division is called karyokinesis when nucleus divides into two nuclei, and it is followed by division of cytoplasm, which is called as cytokinesis. Question 6. The amount of DNA per cell doubles during dash. See the diagram. It is the S phase of interphase when amount of DNA per cell doubles, but the number of chromosomes remains the same. Question 7. Match the column 1 with column 2. Column 1A. Anaphase 1, it matches with 3. In this stage, separation of homologous chromosomes takes place to the poles. B. Telophase, it matches with 4, where reappearance of nucleolus and nuclear membrane takes place. C. Anaphase, it matches with 1. During anaphase, separation of chromatids to opposite poles takes place. And D. Prophase 1, it matches with 2, where synapses occurs, means pairing of homologous chromosomes takes place. So, answer is A matches with 3, B with 4, C with 1 and D with 2. Question 8. What is synapses? See the diagram. The pairing of homologous chromosomes during zygotine of prophase 1 is called as synapses. Question 9. Supply the scientific terms for each of the following. 1. Phase in the cell cycle when proteins and RNA are synthesized. It is the interphase of cell cycle when synthesis of RNA and proteins take place. 2. Phase of mitosis when centromeres split and chromatids separate. It takes place in anaphase stage. क्रोमोसोम का सेंट्रोमियर स्प्लिट हो जाता है एंड क्रोमेटिड्स क्रोमोसोम के सेपरेट हो जाते हैं. Question number 10. What is the duration of cell cycle in yeast? It takes 90 minutes in yeast for completing one cell cycle. Question 11. What is crossing over? See the diagram. Genetic material का exchange होना between non-sister chromatids of homologous chromosomes is called as crossing over, and this takes place during Pachytene stage of prophase 1 of meiosis 1, and enzyme involved in crossing over is recombinase enzyme. Question 12. Name the point at which sister chromatids are attached with each other. The two sister chromatids are attached together at a point called centromere. Question 13. What is the role of kinetochore? See the lower diagram. Kinetochore is present on the sides of centromere and it helps in attachment of chromosome with spindle fibers. Question number 14. Two key events take place during S phase in animal cells. 1. DNA replication and 2. Duplication of centriole. In which part of the cell these events occur? DNA replication occurs in the nucleus, while duplication of centriole occurs in the cytoplasm of the cell. Question 15. Given that the average duplication time of E.Coli is 20 minutes, how much time 2 E.Coli cells take to become 32 cells? See the table and calculate the answer as per the table. One E.Coli cell will form 16 cells in 80 minutes and second E.Coli cell will also form 16 cells in 80 minutes. So, total 32 cells are formed in 80 minutes by 2 E.Coli cells. So, answer is 80 minutes. Question number 16. In mitosis, what would be the consequences if karyokinesis is not followed by cytokinesis? अगर कैरियोकाइनेसिस के बाद साइटोकाइनेसिस नहीं होता, न्यूक्लियर डिवीजन के बाद साइटोप्लाज्म का डिवीजन नहीं होता, then the cell will become multinucleate as shown in the figure. Such a division is called as free nuclear division and led to the formation of syncytium. Question 17. It is observed that heart cells do not exhibit cell division. Such cells do not divide further and exit dash phase to enter an inactive stage called dash of cell cycle. See the diagram. Heart cells में cell division नहीं होता and they exit G1 phase of the cell cycle and enter into an inactive stage called G0 stage, and it is also called as quiescent stage. Question number 18. Why is meiosis called reductional division? Meiosis is called as reductional division because the number of chromosomes reduced to half in meiosis. अगर parent cell में 8 chromosomes हैं, then daughter cells will have 4 chromosomes after meiosis. So, number of chromosomes reduced to half. Question number 19. The cell plate formed during cytokinesis for the formation of new cell wall represents the cell plate formed during cytokinesis for the formation of new cell wall will represent middle lamella in plant cell. Question 20. Differentiate between prophase and prophase 1. See the first difference. In prophase, there is no synaptic pairing of homologous chromosomes takes place, while in prophase 1, pairing of homologous chromosomes takes place. Second, no crossing over or recombination occurs in prophase, while crossing over or recombination occurs in prophase 1. Third difference, prophase is relatively short phase, while prophase 1 is comparatively long phase. Question 21. A bivalent consists of, see the diagram, bivalent is formed by pairing of homologous chromosomes and it has 4 chromatids and 2 centromeres. So, correct option is B. Question 22. Meiosis involves, in meiosis, 2 nuclear divisions and 2 cell divisions takes place. It is divided into meiosis 1 and meiosis 2. So, correct option is option A. Question 23. During Anaphase I stage of meiosis, separation of homologous chromosomes takes place during Anaphase I stage of meiosis and they move to opposite poles. So, correct option is A. Question 24. The post-mitotic phase in which active synthesis of RNA and protein takes place is? Just after the mitotic phase, there is G1 phase of interphase of cell cycle, where RNA and proteins are synthesized. So, here mark B as a correct answer. Question 25. Name the stage and type of cell division represented in A and B respectively. Diagram A. It represents Metaphase I of Meiosis, as homologous chromosomes are attached at the center of the spindle fiber. While in diagram B, it represents Anaphase I of Meiosis, where homologous chromosomes are separated and moved to opposite poles. Question 26. Why is mitosis called equational division? Mitosis is an equational division as number of chromosomes in parent cell as well as in daughter cell is same. Question 27. The cells formed after meiosis I are called? After meiosis I, two cells are formed with half the number of chromosomes as compared to the parent cell, and they are called as dyads. Question number 28. Differentiate between two basic phases of cell cycle. Two basic phases of cell cycles are interphase and M-phase, and the differences between the two phases are: number 1, interphase stage is the phase between two successive mitosis phases, while M-phase is the phase of actual cell division. Number 2, interphase extends for a very long time. It lasts approximately up to 95% of the cell cycle, while M-phase is completed in a short duration, that is, it marks up to 5% of the total cell cycle. Third difference, the chromosomes appear as chromatin material during interphase, while in prophase, chromosomes are distinct rod-like structures. Question 29. Which stage of cell division will you select to study the morphology of chromosomes and why? To study the morphology of chromosomes, metaphase stage is the best, as during this stage the chromosomes are shortest and clearly visible under microscope. Question 30. Label parts A, B, C and D in the given figure. A is centriole, B spindle fibers, C kinetochore and D chromosome. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.